Welcome everyone to chapter 8 and day 11. In this chapter, we're going to talk about metabolism. And this is really our first chapter where we really delve into what really metabolism is, the reactions that occur in a cell and how that sustains life. So let's come up with a working definition for metabolism. And we can call this all chemical reactions within an organism. Okay, so all Now, when we look at these reactions, we can define them a little bit better. We can define them as either catabolic reactions, which is the breaking down of molecules, or an anabolic reaction, which is the building up of a molecule. So, for instance, if we took a dehydration reaction where we took a monomer, say a single amino acid, so this is just a general amino acid, and we added it to a second amino acid, two separate monomers, and they reacted to give us a short protein. So these two amino acids attach to each other to begin to make our polymer. So this is building up of a larger molecule. So we call this an anabolic reaction. So let's write anabolic above it. Now if we took the same protein here and broke it down to form amino acids, individual monomers, we would call this a catabolic reaction. And the way I always remember this is I remember what an anabolic reaction is first. And I think of things like anabolic steroids. And we know that anabolic steroids can be taken to help build muscle mass. And so that's what an anabolic reaction does. It takes smaller things and makes a larger molecule. Catabolic reactions, I just remember that as being the opposite. It's taking down this larger molecule, this larger protein, into smaller proteins. So it's a breaking down of chemicals, of larger chemicals. So those are catabolic reactions. Another thing we need to think about when we think about anabolic reactions is that they will usually consume energy. So let's write consume energy. It takes energy to make a larger molecule. Whereas catabolic reactions, the breaking down of large molecules into small ones, releases energy. I guess I could try to spell this correctly. Releases. And my shorthand for energy when I'm taking notes and I'm needing to write quick is a big E with a lightning bolt. So if you ever see me write that, I mean energy. And same thing here, I forgot to write it, but it is consuming energy for that anabolic reaction. So we just talked about this word energy. So let's go to a new page here and talk about energy. We often think of energy. Your book defines it as the capacity to cause change. I like a simpler definition and just say it's the capacity to do work. When we have energy in our cells, that our cells they will take that energy and do some kind of work with it. So to do work. We're going to talk a lot about energy in this chapter, but let's begin with some common terms, and these are kinetic versus potential energy. Kinetic energy we often think of as being associated with movement, the energy that is stored in the object as it is being moved. And potential energy is energy that's associated with that object's location or that object's structure. So let's draw a couple examples here to talk about this. Okay, my first example, let's draw some sort of mountain or large hill. Okay. And let's draw a large boulder at the top of it. This boulder at the top of this hill, we say it has potential energy. So I'm going to write potential next to this. Okay, potential energy. We say it has potential energy because because of its location, where it's at. It's located high above the ground, and because of gravity, when it comes down here, it's going to come with quite a force. So we say because of its location, it has a lot of potential energy. As it's sitting up there, it has no real usable energy. It just has the potential to generate a great deal of energy. Now, if somebody was standing back here and they pushed this rock, I'm not sure why they would do it, but they pushed this rock, and now this rock comes crashing to the ground, smashes it against the ground. As this rock is falling, because of its movement, we now say it has kinetic energy. And again, remember my symbol for energy, just to speed things up, is a big E with a lightning bolt next to it. So this potential energy is, because of its location, and as it's being pushed off, is converted into kinetic energy, the energy of motion. Now, when it hits the ground here, this rock, would we say it has potential energy or kinetic energy, or really no energy associated with it at all at this point? What we would say is it has no energy at all. The potential energy is lost, and it's no longer moving, so it doesn't have kinetic energy. Now, if we were to take the same rock and take it right back up here, I'm not sure how we're getting it up here, but it's going up here, we would now restore the potential energy. All right, in a little bit, we're going to talk about ATP. And in this ATP molecule, it has a lot of potential energy. But before we go there, I want to talk about a couple other terms that we use when we talk about energy. The first is this term called free energy. I always found this word to be confusing. We often think of free energy. Your book has a pretty elaborate definition to it, but I like to think of it as in the terms of two contexts. The first is that this free energy is the energy, and let's go and write this down, that can perform work in a cell. And another way I'd like to look at it is thinking about where that energy is at. And in cells, that energy is stored in chemical bonds. So let's write energy. This free energy that's going to do work in the cell is stored in chemical bonds. Now, when you read free energy in your book, really this is all you need to know about free energy. There's a lot of equations and a lot of balancing within the cell. And when you move on and you take cell biology and biochemistry, you'll cover this in a much greater detail. But right now, we just want to think of free energy is that unit of energy within a cell that does work. Okay, and we also want you to know that this energy, this free energy, is typically thought of being stored in chemical bonds. Two other really kind of dry definitions that we need to talk about, and they're important to talk about, but I don't want to spend too much time on them. You can read a little bit about them in your book, is an exergonic and an endergonic reaction. So let's write those terms down real quick. So we have exergonic there, and now how about endergonic? So in an endergonic reaction, there are a couple points I want you to know. First, we need to know that there is a net release of free energy. And I also want you to know that exergonic reactions are spontaneous. And what do I mean by spontaneous? What I mean by spontaneous isn't that it occurs really quick, but that it doesn't require energy for this to happen. It's energetically favorable for these kinds of reactions to occur. And that's why it releases energy. It doesn't use energy. And the way I remember this is that I think of EX for exit, and so this is free energy that is being released or being exited by the reaction. Endergonic reactions are the opposite. They absorb energy. And because they use energy in order for them to occur, they are not spontaneous. They require energy for them to occur. So they are not spontaneous. And this is several pages in your textbook, but really this is what you need to know when we're thinking about free energy and exergonic versus endergonic reaction, as well as potential and kinetic energy. And really, um, what we said here on this page here is if you get this, you get most of this part of the chapter. All right, on the last slide, we talked about a lot of terms dealing with energy in a cell and how we measure and what those terms mean. Now the question now is, once we generate energy in a cell and we understand the reactions that occur, what is the work that the cell needs to do? How does the cell use that energy to accomplish certain tasks? And when we think about the work of a cell, we can break it up into three different categories. So let's first write cellular work here. And the three different categories are chemical work, and this is a process of pushing an endergonic reaction, making that endergonic reaction work. So an example of this would be making a polymer. So when you make a polymer, it's an endergonic reaction. It requires energy, and that energy the cell uses to create this work, making a polymer. Okay, the next kind of work a cell does is transport. A good example of this from our last chapter is active transport. So remember, active transport goes against the gradient, going from solutes going from a low, from a low concentration to a higher concentration. Right? This is not energetically favorable, and so it requires energy. And once that cell gets that energy, it allows this transport to occur. Our third category is called mechanical work. And this is physical movement within the cell. And when we think of muscle contraction, this is begins as a cellular event. And so this is certainly a mechanical work. So we're going to put muscle here, and that's for muscle contraction. You know, the beating of cilia in certain cells, that would also be mechanical work. So all of these are examples of endergonic processes that require energy for them to occur. So where does it get that energy? Well, the cells do a pretty clever thing, and they couple the reactions. They couple an exergonic reaction with an endergonic reaction. So let's draw a small example of this. So I'm going to draw over here, let's write exergonic for an exergonic reaction. This could be something like sugar metabolism. So I'm going to write sugar metabolism under here. Where sugar, like a glucose molecule, is broken up. And that, when it's metabolized, it generates energy. So let's just draw an arrow over here and write the form of energy that it produces, which is called ATP. And we'll talk a lot more about ATP in just a moment. Remember, this is breaking down of the sugar molecule. It's a catabolic reaction, remember that. And it's also an exergonic reaction. It generates ATP. Now, since it's generating ATP, we might as well couple that right next to a reaction that requires ATP. And so let's write endergonic over here for an endergonic reaction. Remember, an endergonic reaction requires energy, in this case in the form of ATP. And as it's using that ATP in this inorganic reaction, we produce ADP, the breakdown of ATP, plus the inorganic phosphate, the phosphate that's removed from it. Okay, this inorganic reaction, just to remind you, this would be an anabolic reaction. Remember, something that's going to build something up, but it requires this energy. And now once we have this ADP that is resulting, it now re-enters this cycle, and the exergonic reaction will use ADP and inorganic phosphate, while it breaks down sugar in this case, to generate more ATP. Then ATP is used in this inorganic reaction. So in this way, these two reactions are, what we say is that they are coupled reactions. The inorganic reaction could not occur without the exergonic reaction. It gives it the energy it needs to do its job. Okay, we just got done talking about ATP, and we've used this term a lot. And if you've ever had a biology course, you have no doubt heard ATP used a lot, and you were probably told it's the cellular currency for energy, and that's true. But let's be a little bit more explicit at what it's doing, and so let's answer some questions about ATP. What is it about the structure of ATP that generates energy? So how does it make energy? I'm going to write generates energy. What is it about the structure of ATP that makes it so energy producing? And then we want to talk about how this ATP molecule, how does it cause cellular work? How is it used? And then lastly, we want to briefly talk about how do we make more ATP? Or where does it come from? So let's address this first question. How does this molecule of ATP generate energy? What about this structure? And to do that, let's draw a very kind of generic picture of ATP. We have our adenine base. This is like any nucleotide, just like the nucleotides we find in our nucleic acids like RNA and DNA. Okay, so adenine, the nitrogen base, is bound to the ribose sugar, and then initially we have two phosphate groups. These phosphate groups have negative charges to them. So in general, like if think about if you had two magnets in your hand and you try to push those negative charges together in such a way that they actually were stuck to each other. So that would generate a lot of kinds of energy there. But even that doesn't have too much energy. But now when we add this third phosphate to make it triphosphate, ATP, this third one here, you're forcing these three strongly negative atoms right next to each other. And it doesn't want to do this. But you get them together, and once they're together, there's a ton of potential energy in this molecule because of the way the molecule is structured. And this potential energy is all right here in this last bond. And so I'm going to write PE here for potential energy. And as soon as this is made, there's just a ton of potential energy right here. Once it's cleaved and you slice this off, and it almost occurs immediately, ATP doesn't just hang around in the cell, it's hydrolyzed very quickly, and you end up with ADP. And I'm going to draw this here again with our adenine, our ribose sugar, and now two phosphate groups. And our other phosphate group has been released as inorganic phosphate. Okay. So this potential energy is found in this bond. It is then cleaved, this bond, releasing the phosphate, and that generates kinetic energy. Energy to do something with, energy for motion. So that is why we say ATP generates a lot of energy in the cell. And it's all because of this last bond. Now to answer the second question of how does the cell use ATP to do work, I want to go to the next slide because I think we're going to need a little bit of space to talk about this. Okay, now let's think about this second concept of how ATP is used for cellular work. And so I'm just going to write cellular work here. And as we think about cellular work and how ATP is used, we're going to go back and talk about it in context of chemical work, transport work, and mechanical work. But first, let's talk about chemical work. And so for this, let's just make up a make-believe reaction where within the cell we have substrates. And these substrates are probably proteins. And so we have this square protein that we want to combine with this circle protein. This is a highly endergonic reaction. So we wouldn't expect this reaction to occur all by itself. But what we would like it to do is to form this larger protein where the square and the circle are combined. However, as I said, this is not a favorable reaction. So let's put an X through this. This is not ever going to happen. But what we can do is add ATP, that energy that's added to generate this cellular work, this chemical work. So we add ATP. And what ATP does, as it's being hydrolyzed to release that third phosphate group, let's pretend that this blue substrate, this blue protein, now binds this extra phosphate group, right, like so. When this phosphate binds to this protein here, a couple things could happen. You could have a change in charge. In fact, you'll certainly have a change in charge because this has a negative charge on it. But it could also change the shape of this protein a little bit. Both changing the charge and the shape of this protein, now in this made-up example here, will facilitate this square substrate to bind to the circle one with the phosphate attached to it. And now we can generate our desired product of the square and the circle combined. The phosphate is also at some point going to probably be lost as well. Okay. So again, let's just go over this real quick. We have our square and circle proteins. They won't naturally go together, so we add ATP. ATP is hydrolyzed. The phosphate is added to the square protein in this example. This facilitates the binding of the circle protein because of a change in shape of the protein or a change in charge of the protein. So this intermediate now then facilitates driving this final reaction here to produce our desired product. So this is how ATP can be used to generate cellular work. Okay, on this next slide, we're going to talk about how ATP is used in transport work. And as we talk about transport work, let's draw a very simplified membrane here. And let's say we have lots of sodium on this side of the membrane. And we have quite a bit less sodium on this side of the membrane. But we want to move sodium across the membrane. However, this is not an energetically favorable process, mainly because of the concentration, but also sodium has a charge on it. So it's not going to freely diffuse across the membrane because of the concentration problem and because of the charge. So this is not going to happen. The way we make this happen is that we have a transport protein. This transport protein, let's go ahead and draw it like something like this, can bind sodium. And once sodium is bound to that protein, it just sits there. It can't get across the membrane still. The only way we can get it across the membrane is if that protein changes its shape. And one way it can do that is by adding ATP. ATP is hydrolyzed, generating ADP and inorganic phosphate. And this phosphate now is added to this transport protein. When it is added to this transport protein changes shape in such a way that the sodium that was once bound here is now on this side and can be released on this side of the membrane now. I should also draw the phosphate here as well. So this is how transport work is generated using ATP. ATP binds to the transport protein, it changes its shape in such a way that it forces the sodium onto the other side of the membrane. Our last example is mechanical. And mechanical, as we said before, means movement. So let's look at something inside the cell. And I'm going to see if I can find a short video that shows this as well in just a moment. But let's draw it out here. Let's say you have a microtubule. And just to remind you, a microtubule is a cytoskeletal element found in the cell. Things bind to this cytoskeleton, this microtubule, and move it across the cell. And it does that by having a motor protein. And we'll make the motor protein here red. And as it's moving in this direction, there's a vesicle attached to it. And our goal is to get that vesicle all the way over here. So it's got to move. And the way it moves here, a little differently, but this is a very similar concept. Because remember, it's not going to move by itself. It's an endergonic reaction. And so what happens is that ATP is bound to the motor proteins on this microtubule. ATP is then cleaved, and when it's cleaved, it forces, it's almost like two legs walking, it forces this back arm here to move forward, thereby generating movement. And what's released here is ADP. And then before it can take another step, ATP has to bind to it. So it's just constant ATP binding to the motor proteins. Then the motor proteins with ATP have the ATP cleaved, hydrolyzed, and that energy that is released causes a conformational change, a change in the protein structure of this motor protein, and literally makes it walk along the microtubule. Let's look at this last example here in a short video. So here's a really cool video, and I think we might have shown this in class at one time, showing this microtubule shown here with the motor protein right here and the vesicle attached to it. So this is happening in our cells all the time, these motor proteins just trucking along this microtubule, moving vesicles back and forth. Okay, that's all they do. So let's watch this. And you see how the pieces of this protein are moving one step at a time. Okay, let's watch this again here. I'm going to pause it here. Okay, right here, ATP, it's not shown here, but it would bind right here to this motor protein. That ATP is then cleaved. As it's cleaved, it generates energy to cause this other protein to move forward as it walks. See, watch this. Whoop, there it goes. And it's constantly binding ATP, releasing, hydrolyzing ATP as it moves along this microtubule. Let's watch this again just real quick. Moving it along, binding ATP, releasing ATP, binding ATP, releasing ATP. Constantly doing this as it moves this vesicle to one part of the cell. Okay. It's pretty cool from a nerd perspective, right? That this is happening in all trillions of our cells all the time. These vesicles are moving things back and forth, and we don't even have to think about it. Now let's talk about how we make ATP in the cell. And there's two ways that we want to talk about. And there's only one way that we're going to spend any time on today. The first way is oxidative phosphorylation. And we're not going to spend much time on this at all right now. Oxidative phosphorylation. This is the phenomenon that occurs in our mitochondria, and it generates a tremendous amount of ATP. And we're going to talk about that towards the end of the semester when we talk about respiration and photosynthesis. But for now, you should just know that oxidative phosphorylation generates ATP in our cells. The other way, which is more relevant for this chapter and our next topic of enzymes, so let's talk about this a little bit. And this is called substrate level phosphorylation. So again, we're going to talk about this kind of protein here. It's an enzyme, and we're going to talk about enzymes in the last part of this podcast. But you have this enzyme here. I'm going to draw it like this. And what it does is it binds ADP, so ADP, And it has this substrate protein that's bound into this enzyme. And this substrate protein has a phosphate group on it. And what happens next is this enzyme pushes these two together, this ADP and this P, releasing the ATP from it. And now we can make ATP within the cell. And this enzyme now goes back and binds another ADP and another phosphate group. And then they push it together and then release ATP here. So two ways our cells make ATP, oxidative phosphorylation and substrate level phosphorylation. Okay, now let's start talking about enzymes. This is the last part of this chapter that we want to talk about, and it's probably the most important part of this chapter. It's all important, but this really is an important part, because we're going to talk about enzymes throughout the semester. Alright, so let's have a working definition for enzymes. And we can think of enzymes as catalysts that speed up a cellular reaction. So let's write that down here real quick. Okay, that's my symbol for reaction, RXN. So let's think of an example here. Let's say we had this sugar called sucrose. Sucrose is a disaccharide, so it's made up of two monosaccharides. But let's say we want to take this sucrose and convert it or break it down into its two monomers, which are glucose and fructose. Now this reaction, maybe if we left it go long enough, it would actually occur. But in the time frame that we need it to occur, it's not very useful. So the way we speed this up faster is by adding an enzyme. And this particular enzyme is called sucrase. So I'm going to write sucrase here. But it's just an enzyme that specifically speeds up the cellular reaction converting sucrose to glucose plus fructose. So this enzyme influences this reaction and makes it more likely to occur. It's a catalyst. So how does it do this? That's kind of an interesting question here. How does it influence this reaction in a positive way? Well, let's first draw this little table here. And on the vertical axis we have free energy, and over here we have time, the progress of the reaction. We know up here the amount of free energy. And if we had our sucrose here, I'm just going to put S for sucrose. In order for it to have enough energy to create this reaction, the free energy has to go way up there. But if it did, we would generate our products of fructose plus glucose. So what this enzyme sucrase does is it lowers the amount of energy we need to achieve in order to make fructose and glucose. What we call either this without enzyme or this with enzyme, this space here of free energy, we call this the activation of energy. Or I'm just going to write it here as energy activation. So energy of activation is needed to generate these products. We add the enzyme, it lowers the energy of activation, the amount of energy necessary to complete this reaction. By doing so, it makes this reaction much more likely to occur. So I remember way back when I was taught that enzymes cause these unfavorable reactions to occur by lowering the energy of activation. And I memorized that and all was good, but I never really understood what the enzymes are doing. And what the enzymes are doing is they're binding the substrates in such a way that they allow the substrates to come together, and it facilitates them creating this product. So let me draw an enzyme here. Here's a good friend of ours, the enzyme. And what we have here is, let me draw a couple sites on here. We're just mainly one. This is our enzyme, E. And this pocket here we call an active site. I'm just going to write an AS for active site and draw over here with an arrow. So how does this active site and this enzyme cause a cellular reaction to occur? Well, let's just make some silly shapes here. Let's say we have a square substrate and a circle substrate. And in a particular cellular reaction, we would like those to create a product. And maybe this product now is a square attached to a circle, like so. Now, this reaction does not normally occur, and in this kind of made-up example we're talking about here, it doesn't occur because these two substrates never come together in such a way that allows them to bind. Well, what our enzyme now will do is it will bind the square, and it will bind the circle. And now that both these substrates, the square and the circle, are in that same active site of the enzyme, they're right next to each other, so now it's going to facilitate the two forming a bond between each other, generating this product here. So again, the substrates bind into the active site. Once in the active site, they complete the chemical reaction, and then it is released from the enzyme. And now we have created this particular product that now can go do what it needs to do. Now, a couple other things we need to add to this are cofactors or coenzymes. And a cofactor could just be a chemical. It could be like a methyl group. I'm going to put an M there. Well, actually, let's not call it anything. Let's just call it a CF for cofactor. But cofactors are small chemicals that can bind to an enzyme. They can bind here. They might bind in the active site. But when they bind to the enzyme, they make the enzyme more efficient. So most enzymes have some kind of cofactor that will bind to them. Another term your book uses is a coenzyme, and we'll put that here. A coenzyme that serves the same function as the cofactor, except that it is a protein. It is an organic molecule like a protein. Cofactors and coenzymes, they bind enzymes to make the enzymes more efficient. The difference is that the cofactor is a small chemical, whereas the coenzyme is an organic molecule like another protein that's binding to the enzyme to make it functional. But they turn out to be very important things in the cell that speed up enzymes. Okay, so we just talked about how cofactors and coenzymes can influence an enzyme's activity. But there are two other things we want to talk about that are capable of influencing the enzyme's activity. Okay, I lied. There's a fourth thing too I also want to talk about. Okay. The second and third thing, they're very similar in some ways. Temperature and pH. Both temperature and pH, if you increase the temperature, lower the temperature, increase the pH, or lower the pH, what they can do is they can change the shape of the protein. So if we were to make kind of a generic example here, maybe we had a protein that looked kind of like just a glob. But when you increase the temperature or increase the pH, what you might do, so let's go like this. Let's increase our pH. That will change the shape of the protein. And maybe now, by changing that shape, what we have done is exposed an active site. And now the protein is active. So this is a way we can change the activity of the proteins by increasing the pH, lowering the pH, increasing temperature, lowering temperature. And the result of doing this is a change in the shape of that protein that either makes it more or less active. The fourth thing here is the concept of inhibitors. There are various kinds of inhibitors that when added to an enzyme will inhibit the activity of the enzyme. So how does that work? Well, there are two different ways, and I'm going to draw a line here because I want to keep this all on the same page. The first one is what we call a competitive inhibitor. So let's write the word competitive here. And this is an inhibitor that competes with the substrate for the active site. So let's draw our favorite enzyme over here, just like so. And usually, remember we said we would have a square or a circle that we would want to bind into the active site. But a competitive inhibitor is also capable of binding into that active site. So let's make this kind of a large rectangle. If this binds to the active site, what happens is it will block the active site, like so, preventing the true substrates, the square and the circle, from our example over here, from binding. Okay. Again, this is called competitive inhibition, where we have an inhibitor that mimics the true substrates in such a way that it binds to the active site, preventing the real substrates from binding. Our other example is called a non-competitive inhibitor. I should have left myself a little bit more room, but we'll make do. And in the interest of space, I'm just going to call this non-COMP, but this is non-competitive inhibition. So what happens here with non-competitive inhibition is our enzyme, shown here, looks like this. But there is a site away from the active site, so let's draw it right here, that can bind a non-competitive inhibitor. Let's say it's just this small chemical here. When it binds to this non-competitive inhibiting site on the enzyme, so it binds here, I'm going to color this in green because it just bound to it, and when it binds to this, what happens is it changes the shape of the protein in such a way that our substrates, the square and a circle, can no longer find the active site to bind. It's blocked. So let me summarize really quickly the 4 things that can interfere, enhance the activity of an enzyme. Number 1 over here is a cofactor or coenzyme. They bind to the enzyme and they will increase the activity of the enzyme. Temperature and pH, they will influence the shape of the protein in such a way that it may now hide the active site, or it may open up that active site, depending on the enzyme you're talking about. The gist of this is that it changes the shape of the protein. The last thing are these inhibitors. And we talked about competitive inhibition and non-competitive inhibition. The big difference between these two is where the inhibitor binds. In competitive inhibition, it competes with the true substrates for the active site. So the competitive inhibitor, if it wins out, it will bind the active site, preventing the real substrates from binding. In non-competitive inhibition, the inhibitor, in this case the green square, doesn't bind to the active site. It doesn't compete for the active site. It binds to a separate site on the enzyme. When it binds to this separate site on the enzyme, it changes the shape of the protein in such a way, as we see here, that the real substrates, the square and the circle, are no longer able to bind to the enzyme. Okay, we just finished chapter 8 here. In this chapter, remember, we talked a lot about energy, we talked about work, we talked about how ATP, the energy of cells, causes this work to occur. We talked about the role of enzymes, a lot of stuff in this chapter. And the stuff in this chapter is sometimes difficult to digest. And so make sure you go over that study guide, the note-taking guide, really well. And hopefully you made notes as I went through this podcast. If you have any questions, please make sure you come see myself or Dr. Pangle or Dr. Learman, and we'll be happy to help in any way we can. Good luck, and we'll see you in class. Bye.